Okay, let's talk like cell biology, and for the specific part right now, membranes. We're going to talk about the anatomy of a membrane right here. So this is what a cell membrane looks like. It is made out of phospholipids, and I didn't mention it in the last video at all, but a phospholipid is same thing basically, except that it's made out of a phosphate group at the top and one saturated acid and one unsaturated acid. That's why there's this little like curve going on, and that makes it so the top of the phospholipid is hydrophilic, the head, while the tail is hydrophobic. This, like, having a hydrophilic and hydrophobic part is called amphipathic. There is like unlikely chance that you will actually need to know this, but if you do see it on your test, you're probably going to be able to figure it out on your own. So when you have one here on the top and one here on the bottom, that is called a phospholipid bilayer. In the phospholipid bilayer, you'll have tons of different proteins. For example, you'll have a surface protein, which is when it's like right on the top. You'll have a peripheral protein, which is when it's kind of embedded into just one layer, and an integral protein. There is a few little thingamajigs I've put down here. First, let's talk about the peripheral protein right here, or the glycoprotein. There is a glycogen thing going on right here, and this is sticking out, and the reason for it is it's for like cell communication, right? Another cell can realize, oh, it's one of us because of these peripheral glycoproteins that are sticking out. Now, this integral protein is an integral protein channel. So this thing allows things from low to high concentration to just like, or high to low concentration, get in and out of the cell. And just to make this a bit easier to understand, everything over here is extracellular, and everything over here is intracellular. Okay, so this is inside the cell, this is outside the cell. Now, one thing that you should also know is that there is some cholesterol in here, just a little bit. And the purpose of this cholesterol is to make the phospholipid bilayer less rigid and more like flexible and bendy. Membranes, such as this one right here, are selectively permeable, which means some things are allowed in and others not. Okay. Small nonpolar molecules as well as water can pass through easily and don't require any energy. That's passive transport. And there are three types of passive transport. There's simple diffusion, which is when a molecule goes from high to low concentration straight through. Osmosis, when water goes from low to high solute-wise, or high to low water-wise, straight through. So the way I wrote this down is a bit confusing. Basically, if the cell is saturated with a bunch of stuff inside and its outside isn't, then water will rush into the cell. It's going from low to high concentration of solutes. And on the inverse, if the outside of a cell is filled with a bunch of crap, but the inside of the cell is filled with basically nothing, it will go from low to high concentration of solute and water will rush out of the cell, basically turning it into a raisin. We will get into that in a second. Facilitated transport is when a molecule goes through a transport protein, like this one right here, without using any energy. There are some that do require energy, but there's also some that don't. So, active transport, on the other hand, is when you have a big polar molecule or an ion going through the membrane, and this will use energy. So you can have a pump, like a transport protein that uses ATP, or your like little itty bitty energies to move a molecule through. Then there's exocytosis and endocytosis. So endocytosis is when a molecule is being eaten up by a cell, and that's called phagocytosis. Or if it's drinking things from the outside, which is pintocytosis. You're not going to really need to know that. Just know that endocytosis is when the cell eat something. Exocytosis is when it poops it out, basically. Exocytosis is cell pooping, where a molecule is put in a membrane, shot to the main cell membranes, binds with it, and then lets it go. Right? So it just has this like trash bag that it shoots through the membrane, bonds with the membrane, and then the trash just poops out. Then there's a co-transport. Whoa. Okay, I don't know why I put co-transport so up close. Co-transport is separate from active transport. Co-transport is where one molecule going against the gradient uses the energy from another one going with the gradient through the membrane. So it's still kind of using energy, but not directly. And to an extent, some people argue, oh, it's kind of like active transport because you have to create that gradient in the first place. But, you know, who cares? Let's start with a uniporter, which is another one of your integral protein channels where, you know, your solutes go from high to low concentration. Very simple. Then you have your symporter, which is when you have one molecule or one solute going from high to low, and you have another one going from low to high, and they're both going in the same direction. And it's using this energy from the high one to push it through. That's called a symporter. Then you have your antiporter, where you have one going from... Whoa. Okay, I made a little bit of a mistake, but I just fixed it. In antiporter, one thing is going from... Oh, never mind. I didn't make a mistake. Let me fix that one more time. One thing is going from high to low concentration, and the other one is using that energy to go from low to high concentration. So one is going from high, one is going from low, and they're switching parts in the cell, and that's an antiporter. Let's talk very briefly about tonicity. So things want to go from high to low concentration, as we saw. Tonicity is the ability of a solution to cause a cell to gain or lose water to maintain homeostasis. So that thing that we talked about with the water and its things, let's move this over, is just, you know, tonicity's whole thing. So if a cell is filled with crap and its surroundings are not, the cell is hypotonic, okay? It is, you know, just getting water to go inside of it because it is lacking water. Then you have on the opposite end, hypertonic, where a cell loses water because these seem incorrect. Hold on. Never mind, this is all right. Hypertonic is when the cell has less solute than its surroundings, so water is leaving the cell. Isotonic is when the surroundings and the insides are the same. Now there is an equation for this called water potential. Specifically, there's pressure. The equation is this little trident thing right here, and it is trident is equal to pressure potential plus solute potential. If you're in an open beaker, the pressure potential is 0. You will usually be given the pressure potential. Okay, so there's nothing to worry about in that regard. Your solution potential, though, is negative ICRT. And if you don't know what that is, it's very important that you know now. I is the amount of parts the molecule breaks down into in water. So covalent, because it doesn't break apart in water, is just 1. But if you had salt, that's 2 separate parts that it breaks into. So your I would be 2. Now C is your concentration of your solution. R will always be 0.0821, and T will always be temperature in Celsius plus 273. If a cell is isotonic, that means that the cell's water potential is going to equal its surroundings water potential, which will make it easier to find out these like unknown variables. So your body, just like these cell membranes, wants to maintain homeostasis and always wants to be at a set point, as it's called. So there is a positive feedback, which is like a chain reaction causing exponential increase. So that's like climate change. While the negative feedback is when it's trying to reach that set point, right? So a sequence of events is trying to return to that set point. As it does, it happens less and less. So think you are really, really hot and you are sweating. as you sweat more, you're cooling yourself down, and you're cooling yourself down is making you sweat less. So that's negative feedback. Now, surface area to volume ratio. A cell wants to maintain homeostasis as much as possible. So it needs to absorb and expel stuff as soon as possible. To do this, the cell's surface area to volume ratio should be skewed to surface area, so it can absorb more stuff into its body quicker. Parts of a cell, you may or not be expected to have like a general grasp of each cell organelle, but here are some right now. In both plant and animal cells have nucleuses, which hold DNA, send out instructions, make ribosomes, mitochondria, which make ATP through cellular respiration, which will be explained later, ribosomes, which make proteins with instructions from the nucleus, rough ER, which has ribosomes and makes and moves proteins. The smooth ER, which has no ribosomes, makes lipids and detoxes chemicals. The Golgi apparatus, which receives, modifies, and sends out proteins. The lysosome, which breaks down trash. Peroxisomes, which break down fatty acids and detoxify the cell. Cytoskeleton, which gives the cell structure. Centrosome, which breaks apart chromosomes and cell division, which we'll come back to this later. Cell membrane lets things go in and out, right? We've already talked about that. And cytoplasm, which just fills the cell with fluid. Now, there are some organelles that are only in plants. That would be the chloroplast, which turns sunlight, water, and carbon dioxide into sugar and oxygen. A central vacuole, which is a big storage container filled with water. And a cell wall, which is like a rigid cover around the membrane, which is what gives cellulose which is made out of cellulose, as we talked about, which gives, your lettuce, your celery, all that stuff, that crunch sound. Organelles that are in animals only are vacuoles. Well, they're found in both, but they're more prominent in animals. And these specific vacuoles I'm talking about are like these food vacuoles. They have like a bunch of sugar stored in them. Lysosomes are found in both, but there's a lot more in animals again. And as we mentioned before, lysosomes break down trash. So let's really briefly talk about juxtacrine signaling. So cell communication can send signals to each other in three main ways. There's juxtacrine signaling, when a cell is signaling to another cell that is juxt next to it. Autocrine signaling, when a cell signals to itself, and autobiography is about me. Paracrine signaling, when a cell signals to a nearby cell. A paragraph is kind of long. And endocrine signaling, which uses hormones that travel through fluids, but an essay is much longer. Okay? And if any of you guys know how to count, you'd find out that there's actually 4 main ways to communicate, not 3. When a cell is sending out a signal, the signal is a chemical described as a ligand. These ligands are received by receptors, and these receptors can only receive certain ligands, signals like a lock and key. So, a way to think about that is that you smelling stuff is from little pieces of whatever you're smelling flying off into the air, touching your nose receptors, and when it smells or when it reacts with a specific shaped ligand, it will release the smell of that tastes good, or smells good, or ugh, I do not want to put that in my mouth, right? There's multiple types of receptors. There's your plasma membrane receptor, which is a membrane in a cell, or the receptors in the cell membrane that attach to ligands that can't cross across the membrane. You got your G protein coupled receptors, which is when a ligand binds to this receptor and it changes shape and releases a G protein. Then you have your ion channels, which act as a locked door, and once it receives a key, it opens or closes in response. You have an intracellular receptor, which is basically like the plasma membrane receptor, but inside your cell. Receptor in the cell that can influence gene expression if it receives a signal. Okay, now let's talk about the steps of reception. So, number 1, reception. When a receptor is received by a ligand, or correctly, it would be when a ligand is received by a receptor. Transduction. A signal is converted that brings out a specific cellular response, such as a second signal being sent out inside the cell, Or B, a phosphate group is added, activating a protein, which is called phosphorylation. Or C, the removal of a phosphate group, turning off a protein, which is called dephosphorylation. Then you have 3, eventually, your response, where a transduced signal is triggers a specific cellular activity. So maybe it is apoptosis. Something is up with the cell, and it receives a signal telling it, hey, you have to kill yourself for the betterment of all the other cells. So it will do just that. That is everything I'm going to talk about for membranes and cell organelles. I will see you in the next video. Goodbye, and good luck.